I am Matt Pillar, Chief Editor at Bioprocess Online and host of the Business of Biotech podcast. And I thank you for joining us for today's discussion on therapeutic cancer vaccines. This isn't a new concept, but a newly re-energized concept with a particular focus in the cancer arena. And that's what we're drilling down on today. And I couldn't be happier with the panel of leaders of emergent companies in the space than the one we've assembled for you today. Dr. Peter Berglund is Chief Scientific Officer at HDT Bio, where self-amplifying RNA is the key to its cancer vaccine candidate's immune-stimulating response. And he should know, when Dr. Berglund was at the Karolinska Institute, he, in fact, published the first evidence that an RNA vaccine could elicit both humoral and cellular immune response against the pathogen. That was way back in 1994, for the record. Dr. Berglund previously served as Executive Director of Research at Immune Design, and prior to that led the vaccine division at Liquidia Technologies and led vaccine discovery at AlphaVax. And when I say way back in 1994, Dr. Berglund, I don't mean to date you.
Very good.
RNA before RNA was cool. Dr. Robert Georgantas is President and Chief Technology Officer at Providence Therapeutics, which is working on a personalized mRNA cancer vaccine to treat women with advanced ovarian cancer, with plans to expand into brain and breast cancer. Prior to joining Providence earlier this year, Dr. Georgantas served as Chief Science Officer at Biologics and Director of Immunology at AbbVie. He earned his Ph.D. in pharmacology and molecular medicine at Johns Hopkins, where he also served as an adjunct faculty instructor. And finally, Dr. Johanna Kaufmann is EVP of Oncology at Codagenix, a company that's zigging a bit while much of the industry zags, while it sometimes feels like the entire industry has gone all RNA all the time. Codagenix's breast cancer and solid tumor candidates, dubbed viral therapeutics, leverage immunostimulatory viruses. Prior to Codagenix, Dr. Kaufmann was scientific director at GSK that is, led mechanism of action studies for immuno-oncology assets at Tesaro, and headed three preclinical herpes virus vaccine programs at Genocia Biosciences. Dr. Kaufmann earned her Ph.D. in cancer virology from the German Cancer Research Center and Heidelberg University in Germany. Joe, Peter, and Robert, welcome. Thank you for spending your morning with us.
Thanks, Matt. It's great to be here.
Thanks for inviting us.
Yeah, wonderful to be here and talk about RNA vaccines for cancer therapies.
Well, I'm excited you guys all agreed to spend some time with us this morning, and I want to start sort of at a high level with a conversation around the why behind all of this. As I noted earlier, this is not necessarily a new idea, but a newly re-energized idea. And Robert, I'll start with you since you're largest on my screen right now. Give us your perspective on how the concept of therapeutic vaccines intends to perhaps change the cancer care paradigm.
Yeah, absolutely. I mean, we're at a point now where, we're at the end of probably a 30 or 40 year arc of cancer vaccines, of the idea of using the immune response, the body's actual immune response to treat cancers. I mean, we've known for a very long time that there are mutations that should give new antigens, but we're at a nexus now where we have enough of the biology to really understand what's going on with the cancer, that we can have an immune response against it. I mean, there's been a long history of tumors that spontaneously, go away. It's been thought for a very long time that was due to the immune response. But, a lot of the early work that was done in this area, it failed. And it failed because I don't think we understood the immune system enough. I don't think that we really understood antigen presentation well enough. And I don't think that we had the tools to actually go in and do things like, even precision healthcare to understand what mutational burden, a tumor had. And we have those tools now. So we have tools to do genomics, to find those mutations so that we can better focus our cancer vaccines. We have a much better understanding of the immune response. And more importantly, we have a much better understanding of how the cancer, or at least a large majority of cancers, evade the immune system, things like checkpoint inhibitors. James Allison's, invention of, a PD-1 inhibitor really has changed the landscape of cancer immunology and now makes vaccines extremely viable.
Yeah. Thanks, Dr. Georgantas. I want to remind our audience real quick before we move on. Please do ask questions as we dig into the content here. We'll be taking questions as we discuss the topic. So we're not going to batch them for the end of the conversation. So make sure you're asking those as we go. Joe, I'm interested in your perspective next on sort of the concept, why the concept is ripe for therapeutic vaccines right now. Particularly, you and I talked earlier, and we talked a little bit about the concept of early stage disease intervention versus late stage therapy, and I know that sort of plays into the approach there. So share your thoughts with us.
Sure. I guess I'll start by maybe piggybacking on the comment earlier from Robert. Obviously, cancer vaccines play a role in the larger arsenal of tools that we have in the immunotherapy space. And in a way, while the checkpoint inhibitors that we already introduced sustain a T-cell response that's directed against cancers, we still have to make sure that these T-cells react to the right antigens. And in a way, cancer vaccines can really kickstart here really the right type of response that we then can sustain with other immune stimulatory agents like checkpoint inhibitors or others. So in a way, it is a modality that can maybe be the beginning to immunotherapy regimens and moving that obviously earlier in the disease stage course where patients tend to be more immunological fit can be very valuable and has probably contributed to the resurgence of cancer vaccines that we're now in the midst of, which is exciting to see. I think there's also always has been this dream that anti-tumor immune responses can not only sort of attack a tumor that exists right now, but can then also prevent additional tumors from arising later or metastases from growing out. Right? It's a systemic treatment, whether it's given locally or not. Ultimately, the effect is meant to be systemic. And I think we've seen very exciting data very recently from Memorial Sloan Kettering at Balakanran's group, where we have seen now direct evidence that micrometastases can be hunted down in the context of a cancer vaccine trial, and that it is the anti-tumor immune response that then prevents those micrometastases from growing out, with lesions that were previously visible in scans now going away. And that's really, I think, the promise, the fascination of the modality, but also evidence that we have now the right tools to measure and understand these mechanisms to engage anti-tumor immune responses. Whether we induce them with personalized approaches to begin with, ultimately the response is personalized. And I think that's what we're all here for, to induce.
Yeah. Peter, as I not so gracefully ascertained from the outset of our conversation, you've been in this space for a long, long time. Tell us your perspective on why now, and what maybe what you've seen over the course of those years.
Yeah, of course, a tremendous development over the course of these last 3 decades. We started with cancer tumor antigens and tumor vaccines, really targeting whole proteins that were known to be upregulated in tumors. And with the advance of the Human Genome Project and the sequencing and understanding what genes are, and also with all that, the technology to rapidly sequence and identify mutations that can cause cancer, then that opened up for the promise of improving the targets for these cancer immunotherapies when it comes to vaccines. I would say that one of the promises that the immunotherapy in general and vaccines in virotherapy in particular have is, of course, safety. Cancer is treated with one of the most extreme tools that medicine has, with knives and radiation and chemo and so on, and that really breaks down the patient or the patient's immune system. And I think with the advance now of these more modern immunotherapies, the quality of life for the patient and safety aspects really changed the way many people look at cancer, and I think certainly how people will look at cancer, a cancer diagnosis in the future.